Dzień dobry Państwu.
Hello, ladies and gentlemen. My name is Katarzyna Mucha, and on behalf of the Management Board of Molecure, I would like to welcome you at the quarterly meeting of the company. Following yesterday's current reports, the Management Board wishes to present information regarding entering into a licensing agreement with Ocean Biomedical regarding YKL 40, and will present an update of the pipeline. The presentation will be given by Marcin Szumowski, Chairman of the Board and CEO, Piotr Iwanowski, Chief Medical Officer and Member of the Board, Zbigniew Zasłona, Chief Scientific Officer and Member of the Board, and Sławomir Broniarek, Chief Financial Officer and also member of the board. We have planned the meeting to take around 40 minutes. Firstly, we will hold a presentation, and after that, we will hold a Q&A session. Please write your questions in the chat, and the presentation will be made available on our website shortly after the meeting has ended. Thank you, and I would like to give the floor to Marcin. Thank you, Kasia. Hello, ladies and gentlemen. First, I would like to start in a bit different manner than usually. We'll get back to the presentation in a moment, but let's start with a few words about a book. The book is titled For Blood and Money. It was written by a journalist, Wall Street Journal journalist, and it describes the history of certain blockbuster drugs and drugs who failed to become blockbusters, and also presents the back alleys of the regulation processes, of investment processes, in the case of drugs which became successfully blockbusters. I recently received it as a gift. I read it, and I have to say that it is very inspiring, and it shows us what needs to be done to be able to create a biotech which can be successfully launched, and also how we can make money on that. So the conclusion is that you have to come up with the right molecule and believe in it. The company has to believe in it, as well as the investors need to believe it, especially the early investors, the ones that get on the train early on. So the entities which invested in PTK inhibitors early on, which are currently widely used in the treatment of chronic lymphatic leukemia. One molecule was sold for $20 million to AbbVie, and the second molecule, a similar second in class, so 20 billion, and the second in class inhibitor was sold for 7 billion to AstraZeneca. Why am I saying that? Because we want to create a company which will worth billions and which will have such products. To do so, we need the faith and determination on the part of both the company and the investors, people who want to invest in this venture. To move to the clinical stages and to the market quickly, we need a lot of money. And that is frequently one of the main challenges that we are coming across in the biotechnological sector in Poland. So this is just a form of introduction. I am not getting royalties for promoting this book, but I can recommend it. Now moving on to the presentation, starting with the transaction which we announced yesterday. You probably already know a bit about our YKL exclusive license, our program. This transaction transfers the rights to this compound and also the IP related to the enzymatic test assay, a unique assay which we created in Molecure and patented. it is transferred to Ocean Biomedical. We will be happy to tell you more about it because I'm sure you will have questions regarding this molecule, regarding this transaction. It is a transaction at an early stage. Further development will be carried out by Ocean Biomedical. We are really satisfied that we managed to close on this transaction because this molecule was received by a company, by scientists who have a lot of experience and expertise in this area and have been working on it for years, and they will continue to develop it as part of Ocean Biomedical. I will give the floor to Zbyszek, who will tell us a bit more about that program, about what we've managed to achieve and what we achieved the partner to do with it.
Myślę, że może po tym wytłumaczeniu.
Yes, definitely. I think that after this explanation, it will become clearer that what happened is positive. Positive because the way that it is perceived now is not that positive. The partner, the expert that we entrusted, is Dr. Jack Elias. He is a doctor, a physician, a PhD. He works at Brown University at the moment. He is an expert in pulmonology. He is renowned across the US. He has established his own company. He has antibodies for YKL. And now he can develop small molecules thanks to establishing cooperation with us. Our goal and our target is to develop our programs. We intend to develop them on our own or together with partners. In the case of YKL, we decided that establishing cooperation with Ocean Biomedical will help us achieve this goal as fast as possible. What is this program about? First and foremost, we are the only company in the world that created about 1,000 inhibitors, binders of YKL40. YKL40 is a protein which plays a role in many processes. It is a complex protein in terms of its functioning because it impacts many pathways, many signaling pathways. Those pathways are both pathological, they entail inflammation and fibrosis, and also the process of creating cancers. What is important here is that combining our main inhibitors in OATD3912 modulates the activity of different proteins. This is complex biology. It's complex enough to make us adopt a broad approach in terms of our main compounds, among others OATD3912, and we proved its efficacy in many models. This was possible because, as Martin said, we didn't just sell the patent for the molecule, but also the patent for the screening molecular test. So if the partner wants to take this program further, they have the opportunity to do that. And this program is based on the functional interaction with the YKL40 protein. As this is a protein that binds with other proteins, this molecule has been designed to ensure that interactions with other proteins will be downregulated. This slide shows us what is most important, that is the convergence of interests. Ocean Biomedical is interested in fibrosis, just like we are. We are still interested in this area. We identified OATD3912 as an anti-fibrotic agent. Ocean Biomedical also has other molecules with this potential. Here in our cooperation, in this graph, we show an experiment which proves the underregulation of fibrotic processes. That was what Ocean Biomedical was looking for. OAT3912 is the most advanced compound. It also has anti-inflammatory impact. In our in vivo model, we showed that it inhibits TNF and increases the expression of interleukin 10, which is an anti-inflammatory interleukin. As we have opposites yin yang, we have TNF driven down and interleukins up. This is exactly the profile that we expect from a molecule that is anti-inflammatory. At the same time, it inhibits inflammasome. This is achieved by underregulating the secretion of interleukin 10B. It also showed anti-cancer activity, especially in the model CT36. We can see that the tumor growth has been blocked. When we look at our pipeline and we see this very short line, and it shows us that a program is at the early stage, we already have a lot of data at that stage, a lot of accumulated knowledge at that point. And this is potential for further growth. Here I presented the YKL example, but in every other program at this stage, we have the same amount of data. A lot of researchers, academics are working on this. Let me reiterate to make sure that this message gets through. No one else in the world has developed YKL40 inhibitors that achieve this effect. People who have spent their careers on working on this, such as Jack Elias, decided to cooperate with us to address their favorite target pharmacologically, and that is very good news.
Książki, że
mikrofonu.
już, przepraszam, włączyłem mikrofon.
The mic was off, I'm sorry. And now let's talk about numbers and figures. I would like to leave you with 4 of those at the end of our presentation. And they are as follows: 38, 25, 1, and 0. Sorry, that was 32, not 38. 32, as you can imagine, is the value of the transaction which Zbyszek has just been discussing, and the scientific achievements that we've transferred via a license agreement to Ocean Biomedical. 25 stands for the value of the market for which we are targeting, which is our priority with regard to the value and the company's business development direction. 1 stands for the number of KIT1 inhibitors in the clinical development. We are the only company in the world who has developed such a molecule up to the stage of clinical development, and it has a huge potential, which we will discuss later on, and it is far more advanced than YKL40. And 0 is the number of patients with MASH who have a double genetic mutation on 2 alleles, where in those patients, so 0 patients of several hundred who were subjected to studies were progressed in the disease. So none of them had disease progression, which makes us quite confident that if pharmacologically we achieve the same thing which Mother Nature has given to the patients, so to this 5% of patients who are biallelic, then we will be successful. This is of key importance in the data that we've collected, and this is why we made MASH the second priority of our company's development. As mentioned at the beginning, to be able to develop projects in clinical stages quickly. And everyone knows that the highest value inflection point is when we manage to demonstrate a molecule's effectiveness in phase 2 clinical trials, and this is quite expensive. So the largest potential, where we've observed the largest number of scientific breakthroughs, in where we understand how the molecule functions in the human's body. Therefore, we decided to stop or suspend molecules and, if possible, sell molecules which are no longer our priorities at this point, because the majority of the funds which we have at hand and which we managed to obtain is what we want to allocate to our priorities, so that we can demonstrate preliminary results of clinical trials for OATD01, if possible, in both indications. So we have stopped cooperation with the University of Michigan. This was a know-how license. To be honest, we didn't pay anything under this license, and we simply closed the program with regard to any procedural aspects without any costs to the company. The results which we obtained were not sufficient for us to be confident that we should continue investing in this program. Therefore, we decided to close it. A second program, which was temporarily suspended because it is very interesting. It is the USP21 program. We had some small breakthroughs, small successes in this program. However, so we would like to be able to return to this program if the financial situation of the company and the available resources allow that. We would definitely like to return to it. But YKL-40 was transferred to very good hands, on very good terms, very satisfying terms to the company, given the fact that we didn't have the resources to develop the program in-house in an efficient manner. I might have skipped one slide too far. Most of you probably had the chance to see this data and saw this presentation, but we would like to underline why this market is so important to us and why the data which we've obtained over the last 1.5 year, either individually or in cooperation with partners, makes this, next to sarcoidosis, the second priority of the company with regard to clinical development. It is a combination of the fact that the data for MASH, especially genetic and metabolic data, which indicates how we reverse metabolic processes by administering our compound. This all makes us believe that we believe this compound should work. If it blocks hitinesis and does it so effectively, and a lot of data corroborates that, then definitely a therapeutic effect is very probable both in MASH and sarcoidosis. After achieving proof of concept, we could possibly also address other medical areas, but currently our focus is on MASH and sarcoidosis because this is where we have the most information, the most actionable data, and where achieving results is most likely. A few words about the company which managed to register and sell drugs for MASH. This is Madrigal Pharmaceuticals. It is a registered company registered in the US. After publishing the results of its third phase clinical trial results, the value of the company jumped by jumped 4 times. The drug is already available on the US market. It still hasn't received marketing authorization in other regions of the world. It is a company which focuses on this disease, on MASH, and it focuses on one molecule which received the marketing authorization. This has, in a way, motivated the market and increased the optimism with regard to the possibilities of treating MASH, because before this compound, Madrigal's molecule, was approved, other clinical trials were unsuccessful and the authorization was not granted. On the slide, you can see the key players. Obviously, we had some interactions with all of them. Most of them are large pharmaceutical companies. Madrigal is rather a smaller company. It is a one-asset company, actually. The growth dynamics, we've talked about it many times, so I won't be underlining it, but of all the potential indications for OATD01, this is the most promising one. How does resmetirom work? How is it different from our approach to treating MASH, to our hypothetic approach to treating MASH at this point? Resmetirom reduces inflammation of the liver and, in a way, fights the steatosis of the liver. So it targets the middle stage of the disease. When it comes to the epidemiological target, this is quite an even distribution. There are a few million patients in each stage of the disease, and when we get to the most severe stage of the disease, there are fewer. The patient population is lower because at this stage the anticipated lifespan is quite short. Our mechanism is different. We have an anti-inflammatory and anti-fibrotic effect. This is what we aim at. And this is a different action because it affects cell metabolism. It can indirectly address the fibrotic process in the liver. Our studies have shown that we can inhibit this process or sometimes even reverse it. So this is quite an interesting area where we could be developing, because Madrigal targets only some of the patients with MASH, and its action is not always optimal. When it comes to the plans for clinical development of OATD01 in phase 2 in MASH patients, we have been analyzing the need to maybe administer this drug on top of Madrigal's drug, and then the drug-drug interactions are of key importance because we do not block the key cycle in this process. We are counting on the fact that no drug-drug interactions would take place. When we did TDI studies before the IPF at an early stage, those studies did not suggest any drug-drug interactions. And the last bullet point on this slide shows us how our compound impacts the liver. So we reduce the influx of monocytes from the blood into the liver. That makes the liver to return to homeostasis. And we observe an anti-inflammatory effect. In more advanced stages of the disease, sometimes it is also possible to reverse the fibrotic changes. Right now, it is a hypothesis which we want to test in the phase 2 clinical trial. This is a slide summarizing what I've just said and what was presented at previous meetings. One important detail is that such a clinical trial, phase 2 clinical trial in MASH, the estimates are that it will cost between $20 and $30 million. And we are talking about a proof of concept study, not a multiple site trial. We are talking about a significant amount of money, an amount of money which biotech companies, not just in Poland, would have trouble collecting. So we are carrying out a number of talks about solutions where we would carry out efforts, and an experienced partner with experience in metabolic diseases who has already carried out such tests. This partner would launch this trial as soon as possible and would carry out in parallel to our efforts, parallel to our KITE sarcoidosis trial.
Let me add that it is not possible to gather more preclinical data to go into MASH. Marcin said that we know how our compound works in this disease, but this research was conducted not just at our company, but also at Galapagos, at different CROs, and independently by academic centers. And our molecule was part of the model. That's why we're so convinced when we talk about this. If we had more experience in MASH or metabolic diseases in our company, if we were capable of obtaining more capital, then probably we would like to do this on our own. As we've said, the highest jump in value is generated when we show efficacy in the second phase of the clinical trial. What is achievable to us is working with a partner to do this, and therefore we will have to share. What is most important to us is time. This market is practically a virgin market, apart from one US-approved company. We want to enter this market as soon as possible by about 2030, when it will be worth several billions. Time is important if we want to do something big. To come back to the analogy with beta-K inhibitors, they received the first approval after 6 years from starting the project. That is very rare, but it is feasible, as proven. Let me hand over to Sławek. He will give you the figures. Thank you, Marcin. Yes, I will be talking numbers now. Q3. It does not reflect the developments that Marcin discussed, that is the YKL-40 transaction and the license he talked about. It will be pleasant in terms of accounting. As you know, the financial value, the balance sheet value, is equals 0. We'll be waiting for upfront payments, then potentially $32 million. We will be happy to carry that in our balance sheet. Coming back to the consolidated results for the 3 quarters, as you can see, we have a drop in revenue as compared to the previous year. This follows from fewer grants that we received in this year. Let me point to other revenues. Those are operating revenues, that is about $500,000. This will grow. This is an allowance that we received for our staff. Since we don't receive the grants we wanted to, we're looking for other tools and mechanisms to generate revenue. We have also some additional revenues, a grant that we received from NIH. It is for anti-fibrotic effects. Our costs are kept in check. They are about 20% lower in terms of general and administrative costs, especially in early phase projects. And our EBIT is comparable year to year. Looking at our cash flow, the left side of our graph shows that we spent about 35 million zlotys. About 13 million have been capitalized in the balance sheet as expenditure on clinical trials, and about 21 million loss reported for the 3 quarters. 15 million of that are expenses on early stage programs. To sum that up, from a cash flow perspective, from the perspective of expenses on programs, we see that spending has slowed down. We signaled this by showing a slower pace in clinical expenses. These are presented here very clearly. We can see that the drop is about 22% year to year, which means we still have 28 million this year, and we have a headcount of 105 employees. Let me add one more thing that I need to emphasize. Marcin talked about these 4 numbers. I'll refer to the first one, 32 million. It's not reflected here yet. Maybe you can see it in the figures. The pipeline, where we are capable of taking economical decisions and suspending those programs which are not promising, of course, those programs also are reflected in the numbers. This trimmed pipeline will give us savings of about 13 million until the end of this year. So let me add that number to this package.
Marcin, ponownie prośba o włączenie mikrofonu.
I'm sorry, the mic was off again. So, to sum up, in a moment we will move on to the Q&A session. But before we do that, let's use an analogy of the book, which I've been promoting so hard. So, the story of the BTK inhibitors was that they were found somewhere in some database of a small seller company which was carrying out some small analysis. Someone noticed it, put it into the pharmaceutics company, which was undergoing significant troubles with its clinical trials, which was rejected by FDA 5 times. And then they decided to start developing this molecule, which they bought for really small money, for peanuts, and then it became a blockbuster. After they've managed to demonstrate incredible effectiveness in leukemias. So starting with CLL, they then had more than 10 more indications in hemato-oncologic diseases. So this was this rough diamond that someone found, maximized efforts, brought together investors, the funds. By the way, the CEO of that company had no experience in biotechnological development. He came from a completely different sector, and yet we had this success. As the second BTK inhibitor was also somewhere there on a shelf in the Netherlands. It was owned by Merck. When Merck was closing its activity in the Netherlands, it sold this molecule to a small startup. You are giving too many spoilers on the book. So they sold it for $1,000. Then Astra bought it for millions of dollars. Coming back to our chitinazes, we are absolutely convinced that Kite1 is this diamond which basically we didn't just find it, we discovered it, but we did it nearly took it out from Yale University nearly 10 years ago, and now it is a potential breakthrough drug which can potentially create a value similar to that achieved by BTK inhibitors if we manage to demonstrate phase 2 clinical trial results. This is possible. Thank you for listening to our presentation. Piotr wasn't taking the floor, but I'm sure he will be answering a lot of questions. Yes, I think it is very likely. Thank you very much for giving this presentation. And now we are moving on to questions.
Transakcja.
Let's start with a question about the transaction you announced yesterday. Let me combine two questions. What convinced you to enter into an agreement with Oceans Biomedical? What advantages, what competencies does this company have? Why are you convinced that they can further develop YKL-40? And the second question, are you pleased with this transaction because 24 million is the amount of grants itself, but does it cover all of the costs? And I'm sure there will be a question about verifying the investment outlays. I won't comment on the amount itself. I'll leave that to Sławek. Zbyszek was pretty convincing, I think, when he said why we chose Ocean Biomedical. It's not a large company, that's true, but it has a very experienced team from many pharmaceutical companies, Gilead among others, and the best scientists and the best resources. As a result, they're moving into territory that is well known to them. I'm sure they will select the optimal development pathway for this molecule. Of course, we engaged in talks with other companies. However, no one else in the world has the same knowledge about this target and about what can be done by blocking this target. Other companies rather expected that we would develop this to an IMD-ready level. As a small company, we must stick to our priorities. This was not an expense that we were ready to incur, to take on the whole preclinical development stage. It made sense to establish cooperation with this partner. Of course, it made sense. 24 million spent on Sławek will comment in a moment. But what do we have here? We have a program where we have tests that can be further developed. We have a molecule that has been proved efficient in vivo models. This is a palpable value. We have a strategy in our company, what we develop and what we do not develop. We focus on USP7 as concerns Michael and our corporation. We want to develop this further, and we think that a partner in the US is the right way to go. Sławek will comment on what has been spent and why and so on, but what I find important is to see the potential and a specific product that has been created from the very outset of this program. It tells them that we have an innovative drug and we achieve an in vivo level with different indications, with a well-analyzed compound that is ready to be further developed. And this is a specific product that we created that can be further developed. That is some serious value. Let me add 2 sentences. We are very pleased. As I've said, the balance sheet value is 0. From the scientific perspective, this partner is valuable. We have people from Yale, Brown, from Merck, from Gilead. These are people who can further develop this drug. We are also pleased with the amounts we have negotiated. As Zbyszek said, this is a preclinical stage program. The 24 million, how was that calculated? I don't know. But we have spent 9 million Polish zlotys net. What we're not pleased with is the reaction. We had a positive announcement and the reaction was negative. Thank you for these answers. Those 24 millions was the grant amount. Let me mention that the company did not use the whole grant. The company informed in October 2023 that you terminated the agreement with the National Center for Research and Development, and the unused amount was about 12 million Polish zlotys, if I remember correctly. Thank you.
Now, one more question regarding the same compound. What is the next milestone in the development of this molecule? What are we waiting for? It is a molecule in an early stage of development, so it's about nominating the drug candidate. And now the question regards the KITE study for OATD-01. How many patients do you have at this time? And is moving the reading of the interim results from the first quarter to the third quarter results from the poor enrollment to the trial. With regard to enrollment at the early stage of the trial, this is something we've informed about previously. What I can tell you now is that definitely enrollment has become more active. The sites have become more active both in Europe and in the US. And therefore, those timeline assumptions which we presented remain valid. However, it is not common practice in the world that we count every patient, especially with trials of this scale, especially in the case of blinded trials. Also, these numbers are changing every day. What I can say is that we are still at an earlier stage of the enrollment. This is still not the level of number of patients which will allow for reading of interim results. We also thought about implementing corrective measures which would increase the enrollment pace in the KITE trial. When do you think those corrective measures can actually give results? So maybe, Piotr, would you like to add something? No, nothing to add. The situation is very dynamic, but yes, additional actions are being taken. With every week, we are taking more actions, not just us, also the CRO company, which is operating this trial. But a lot of actions are taken directly by us, by the sponsor.
Dwa dosyć podobne pytania.
Thank you. We have two quite similar questions now. The question is about the molecule, which is the essence of the business. Galapagos paid 25 million up front, about 300 million bio-dollars for this molecule. What are your expectations concerning MASH, which is a larger market? The previous partnering agreement was for 25 million upfronts. Will the next agreement, taking into account the more advanced stage of clinical trials and the larger market, will be twice as high in terms of the upfront payment? Is that realistic or optimistic? This is a multi-dimensional question. And let me give you a multifaceted answer. The first answer is yes, the Galapagos transaction is a sort of anchor. We're using that anchor in our negotiations and talks. As concerns what happened between 2020 and now, what is happening now, on the one hand, the molecule is more advanced. It has reached phase 2. We have conducted a whole series of tests that have improved its substantive value. We have reduced the risk and increased its efficacy. However, the market has changed, and it's going not necessarily in the direction we want it to be going. In 2020, we had lots of transactions, high upfront payments, and often these transactions were performed earlier on. Now the global atmosphere in the market is that most big companies are waiting for phase 2 results, and that's a catch-22 situation. Of course, if we could take the trial to phase 2, we could expect twice or thrice as high amounts. We will rather do this together with a partner, as I've described earlier on. To us, the upfront value is not the most important thing. We want to enter the market as fast as possible and have the highest possible upside. Even if we get 30, 50 million in an upfront, then it is a one-off fee, while the upside, the milestones, that is important to determine the value inflection point for us. And those are potentially billions downstream. So it's not that much of a bet. In the case of Galapagos, the upfront was important because, well, they shut down later on. But the stakes are high in this game. We believe this should work. We believe that this will work. We will negotiate to sign the agreement as soon as possible and launch the MASH trial as fast as possible. That's priority number 1. And priority number 2, that's what you asked before, how to reach interim analysis as soon as possible. And this burden rests upon Piotr's shoulders. That is his primary task and the challenge that he is coping perfectly well with. And I hope that we will not inform you about prolonging any deadlines in the future. Thank you. A financial question now. By the end of 2025, we're missing about 70 million in funding. Owing to the stock index, it would be difficult to receive funding from selling stock. Would it not be better to freeze certain projects, for example, mRNA and USP7, where you are planning to spend about 40 million Polish zlotys? Let me just add that you have the answer in the right part of this slide. Our decisions are economical. We have taken such decisions for some early stage programs. These are internal decisions taken by the management board. We will take them internally. At this point, no such concept has surfaced. We want a balanced pipeline that will help us achieve what we're showing here. That is the clinical programs with our flagship D01 program. We also want to have something up our sleeve. We want programs in the pipeline when we find partners for our clinical programs. Let me add that apart from the figures from the financial data, companies are also made up of people. OATD01 was discovered and developed by specific people. They have taken it to the clinical phase. They have introduced it into clinical trials. They have taken it to phase 2. Of course, in some models, we trim our company. We leave just 3 people in charge of something and see whether it works out or not. But we haven't reached that point. D01 has such vast potential that we want to continue our trials. We want to continue operating as a company that has more to offer than just one molecule. However, at this point, that one molecule is key to ensure monetization. So yes, we are making savings, reducing, we're not shutting down. Yes, that is not our business model. We have an mRNA grant. mRNA is not costing us much. We have 75% of co-financing. We have an ingenious, brilliant team working on this. We're counting on monetizing that program sooner than later. So D01 is now our first and foremost priority, but it's not the only priority of ours.
Thank you for that. We have some more questions that regard the OATD02 clinical trial. Is there any new information regarding the 20 milligram cohort and regarding the urafic acid? And the question, when can we expect information data on those 3 additional patients? And when can we learn about new decisions regarding the trial? And let me read out one more question, which is quite similar. What, according to the company, are the most likely scenarios of continuing with the dose in the OATD-02 trial? Is the 20 milligram likely to be the final dose, or is it possible that escalation to 30 milligrams is possible? And are subsequent patients in the 20 milligram cohorts being administered the drug? The sites are very active. The trial is going well. Currently, we have those 3 additional patients in the cohort. Another patient is being administered the compound. He has malignant cancer. We are in the mid point of observing the possibility of limiting the toxicity of the drug. Nothing has been observed, and currently in the screening phase, we have another patient who probably, if everything goes well, if all the criteria are met, who next week will also be administered the study drug. Regarding the future scenarios, obviously it's first in human. We find the possibilities of both escalating and de-escalating the dose. Everything at this stage is possible, especially in clinical oncology. What is the most likely scenario? This is something we can answer when we have all the pharmacokinetic and pharmacodynamic data regarding safety of the compound. I personally can speculate that this is probably just a stopover towards further escalation of the dose, but we will see what the more comprehensive data for the entire 6 patients in this cohort will show us. Coming back to the question regarding uratic acid, so our additional biomarker, safety biomarker, the data we are receiving on a regular basis do not suggest anything that would be disturbing. Nothing suggests the need to stop escalating the dose. Thank you for this answer. And can we get a direct answer? When is it possible to expect data for the entire 6 patients? Is it possible at the turn of this and next year? Yes, at the turn of this and next year, it is possible to expect this data. Now, a question regarding MASH. What other steps must be taken by the company or a potential partner to initiate trials in MASH? How long might those efforts take, and what funds might be needed for phase 2? For sarcoidosis, you've got about 100 patients and 30 sites, and something between 60 and 65 million Polish zloty. With regard to any regulatory aspects, I will give the floor to Piotr at the end. However, something that might speed up our actions is that we have already authorization to carry out trials given by FDA to carry out the trials in sarcoidosis. So MASH should go quicker. When it comes to the estimations, we still don't have it, but we are expecting more than 20 million, maybe 30 million US dollars. The population size will in the trial will probably be similar. The drug will probably be administered for a longer period than in the case of sarcoidosis, which is 3 months. The study protocol will depend on our partner. How long might that take? As soon as us and the partner manage to take care of the regulatory aspects and manage to draw up the study protocol, this usually takes into account waiting for the biopsy results. This is something that's not used in the sarcoidosis indication. We could also like to, we should probably have a look at the obvious pathway that was followed by resmetirom as it was being developed clinically. There, in phase 2 trials, that took around 8 months. The sponsor took 1.5 years, more or less, to carry out the entire trial. From a purely regulatory perspective, as the chief of the clinical department, I am looking at this with quite a lot of confidence. We would be, so both us and a few other sponsors are treading on much more stable grounds. So both FDA and EMA are on board, and we already have a drug which already paved the path. So now we are operating on much larger certainty than in the case of our efforts with sarcoidosis. Different endpoints are adopted. We selected an endpoint, and this is something which we discussed previously. We selected an endpoint which is considered quite innovative, but which is shown in articles in literature, which gives us the largest chances of demonstrating the results well. This endpoint allows us to show whether the compound is effective in pulmonary sarcoidosis. And also, based on other trials carried out on sarcoidosis, we know that our primary endpoint, which is based on PET-CT, this allows us to observe the activity of, and other endpoints do not allow this. So all in all, I am looking at the possible clinical development of the molecule in the MASH indication, where we can be far more confident in our actions.
Pacjentów dostępnych, bo to
And of course, the large amounts of available patients. In sarcoidosis, we're talking about 100,000 patients, and in MASH, we're talking about tens of millions of patients. The scale is completely different, so is the availability. The group we are testing in sarcoidosis has been selected in a specific way. It's a subset of all these sarcoidosis patients. This is another level of the challenge we're embarking on. Therefore, enrollment is slower than we expected. Thank you. And another question about Novartis studies in sarcoidosis. How do you evaluate the results? It is not common practice to comment on the results of other sponsors. To the best of my knowledge, unless there are some very recent announcements, the sponsor hasn't commented. Novartis refrained from commenting on their results, so we don't want to do that. Of course, we're analyzing the results. Indirectly, let me refer. In the previous question, I referred to this indirectly. What we see when we analyze the results, we look at particular endpoints, and this doesn't directly impact the clinical development of our molecule. However, each and every result of the scarce research on sarcoidosis, to some extent, expand our knowledge, the knowledge of our company and all the other players, as well as the academic environment in terms of pulmonary sarcoidosis. we gain more knowledge about how to structure and how to perform clinical trials.
Dziękuję bardzo. Kolejne pytanie. Jaki jest potencjał?
Thank you. And the next question, what is the OATD01 potential as compared to Madrigal's registered product? What is the safety profile and the activity profile of the registered molecules? Are these competitors or can they be used in combination therapy? I will be very pleased to take this question. Let me start from the last part of the question, combination therapy. Of course, this is open to speculation. It cannot be ruled out that if the efficacy in some spectacular way would be increased in a combination therapy in the future when both drugs are already available on the market in those indications, well, that would be possible, but we have to bear in mind efficacy. Efficacy is even more important for regulators, and what is even more important is safety. These drugs target the liver. They target a sick liver. Therefore, safety in terms of toxicity is one of the basic elements of drug safety. That's so much for an introduction to my answer. Of course, such considerations are just pure speculation at the moment, but we are looking into resmetirom, into its properties, into its SPC. It's available on the American market, and we see two safety signals. One of those safety signals is the capacity to target solid tumors. It can be used in gallbladder and the gallbladder duct inflammation indications. It has been noted, not to a level of concern, but it has been noted that it leads to an increase in liver enzymes. And in its SPC that has been noted. At this point, when we compare the safety profile, when we compare OATD01 with resmetirom, our drug seems to be different, which is good news. It can be used as a complementary drug or part of a combination therapy in the future. At this point, from the perspective of drug safety, we don't see anything that would limit us. Marcin has already mentioned the profile of the drug in terms of interacting with other drugs. It's also different, which we see positively. In a nutshell, we see it as a complementary drug in the future. That is our medical perspective, and the market perspective, well, it's open to speculation. Let me give you some statistics to tell us about the structure of the market in terms of disease progression. Madrigal's drug targets about 40% of the market, while phase 4, which we would add, that would target 75% of the market. So we would have 3/4 of the market in common. Thank you. Another question about Madrigal, among others. Has the company discussed OATD01 potential in MASH with companies, with big pharma companies, with Madrigal, among others, since the company is comparing its solutions with resmetirom? Let me give you a diplomatic answer. We are holding talks with practically all the companies that are active in this area. It is therefore highly likely that those are most of those companies, perhaps with the exclusion of smaller and less known companies. Most of them are within the range of our talks and negotiations, and these talks are at different levels of advancement. Thank you for that answer. We have a question about a human potential. You've discussed potential limiting of resources. I understand that is about letting some of the staff go. Are you considering any such solutions? Have you made some final decisions? Let me just add that if you would compare the periodic reports we had 113 reports, and now we have 105 members of staff. I think that is a precise answer to your question. Thank you.
The next question regards MASH. A potential partnering agreement in MASH development. Are you expecting an upfront payment, or would you be focusing on royalties and the possible covering of the incurred costs? So a question regarding the terms of a possible partnering agreement with MASH. Can co-development result in a low amount of upfront or no upfront fee? Again, my answer is it depends on who we end up with signing the agreement in the end. But all options are being considered. Obviously, it depends on which partner becomes our actual partner. So some type of upfront fee is something we do expect. But what we also consider is a form of co-development where our company would co-finance the trials in some extent, but the direction we are headed at is that we develop sarcoidosis together, but the partner is taking over the MASH indication with regard to carrying out the trials. And the partner would take over the costs of carrying out trials in MASH. To take this question a bit broader and to make a reference to what I said at the beginning, so they need to believe in the value of our compound and believe in the possible success and in the data that we present. Our main shareholder is fascinated by this project and convinced of its value. And this entity declared that if it was a formula that the company would, if our company was to co-finance the trials, they would be willing to cover some of those costs. So we are enjoying a lot of support from our main shareholder in this regard. Another question regarding the YKL40 and the construction of the trial. This deal is designed in this way that the upfront and the subsequent fees is a mix of fees and stock. So the value which will be expressed in stock will still be determined, and the risk of the changing value of the stock is on our part. So the shifts in the value of stock is more dynamic in the States. Everything happens quicker in the States. We were happy to hear that the stock value of Ocean increased by 30%, and we will be even happier about this fact when we do hold their shares. Thank you. We have one general question regarding the current situation and the moods regarding entering into partnering agreements. Is Big Pharma in general open and determined to enter into such agreements? Is the current situation more positive in the worldwide, in the global scenery with regard to entering into partnering agreements? We are observing an improvement in this regard, but when it comes to Big Pharma companies, this improvement is, this growth is the slowest. They are less, so Big Pharma companies are less willing to pick up programs at an early stage. With later stages of development, they are more willing to enter the scene. I've already said that big companies prefer to pay more. They prefer to pay several billion dollars for a de-risked product in a way. And this applies to all big pharma companies. There are some exceptions, but compared to the period, let's say 3-4 years ago, when there was where we were observing a biotech boom, it is a more difficult process to and time-taking process to close a deal. But the situation is more positive. It is improving. The processes are becoming dynamic, and we are in talks constantly. So in the past it was better, but it's not bad now.
Dziękuję bardzo. Tutaj dosyć ciekawe pytanie.
Thank you. Quite an interesting question now. Could the main stakeholder become the main partner in developing MASH, for example, in a joint venture? Could it take up stock issued in a new issue? As concerns the second part of the question, yes. And the first part of the question, it depends. It depends on the structure of the transaction and whether such a need arises. It was rather a declaration of trust and the conviction that there is vast potential. Of course, thank you. And a question to round up, a general and quite long question. At which point does the company believe that signing an agreement on MASH would be optimal? Which potential contractor could risk investing in the project at an early stage without waiting for full results? I wanted to make sure at which stage, at earliest, it would be possible to sign a partnering agreement. Would it be possible at this point on the basis of the gathered data and scientific knowledge? Or perhaps we have to wait for interim results, or we have to wait even longer? Certainly, when we talk about the possibility of conducting a transaction, it is already possible. It's possible now. In terms of optimizing value and considering whether it's worth waiting a bit longer, well, we need to adopt two perspectives here. If the price is too low, if the transaction parameters are too low, well, we wouldn't want to give up our most valuable asset on those terms. The second factor is time. And I'll keep repeating how important time is. Every molecule has its life cycle, its patent protection period. And we need to optimize this transaction in time to make sure we reap the greatest benefits. It's better to act sooner than later, even if the payments are smaller at the earlier stages or co-financing is lower. Therefore, time is of crucial importance. However, there are multiple factors that we need to balance, and we need to take into account when taking our decisions. It's not a single dimension we're looking at. It's the upfront is not decisive. To us, time is of utmost importance. Thank you. We have exhausted all of the questions. A big thank you to all the listeners for typing in their questions. Thank you to the Management Board for the presentation and providing answers. We encourage you to contact our Investor Relations Department. A recording of this meeting will be uploaded to YouTube, to our channel, and the presentation deck will be made available on our website shortly after the end of this meeting. We would also like to thank you for this meeting and invite you to participate in our MASH and other journeys. Thank you and have a nice day.